Hello, this is a Brighter Futures film about how research improves policy and changes lives. It's a great pleasure to welcome Nick Aldred from the School of Life Sciences here at the University of Essex, marine biologist with expertise in biological adhesives, glues in other words, animals that can stick to rocks and their applications of those glues. So Nick, welcome. Why don't we start with larvae, larvae that are your kind of interest and expertise?
Yeah, I suppose I spend most of my time on very kind of applied science, you might think of it, which I'm sure we'll come to later. But fundamentally, I'm interested in invertebrates, larval ecology. So invertebrates are things without backbones. And there's a fundamental difference between how invertebrates go about their lives on land and in aquatic systems, in the ocean particularly. So on land, things tend to move around a lot. So adult insects and slugs and snails and everything else you might think of, they all move around, and they do that to find food and to reproduce, to avoid predators and lots of other reasons. In the ocean, they can stick to surfaces, which is something that things don't tend to do on land, because in the ocean, the water can bring food, it can take the waste away, it can disperse their kind of reproductive spores and eggs and sperm and all the rest of it. But this presents a problem because animals need to be able to disperse to occupy new areas. If anything bad happens in a local area, they can go extinct. So if we think about things like corals and sponges, barnacles, mussels, all of these things that we know attach permanently to surfaces in the ocean, they all need a way of distributing themselves around the oceans and colonizing new areas. And they do that through larval dispersal. So on land, snails, for example, they'll lay eggs and they'll hatch directly into small snails. In the ocean, most snails will have a larval stage. So they'll release those into the ocean and they'll drift off for hours to weeks to months and colonize new areas. That's a kind of basic.
Yeah, and so that colonization involves being able to stick to a place.
Absolutely, yeah.
And it's difficult to do that in water.
Yeah,
or it requires a different kind of chemistry and physics to go with that. So tell us what happens.
It's difficult for humans to be able to do that in water. We have a range of glues that they're all pretty poor when you compare them to glues in the natural world. So we have things like superglues, cyanoacrylates, epoxy resins, PVAs, and all of these things have been around for ages. They don't work well underwater and they don't work well with contaminated surfaces. So you think the first thing you see when you read the instructions on a bottle of glue is make sure the surface is clean and dry, which natural surfaces never are.
Clearly can't do, yeah.
Yeah. So, so my research focuses on things like barnacles and mussels, particularly barnacles. They're my kind of lab pet species, figuring out how they attach for the purposes of trying to produce biologically inspired glues that could be used in human surgery or any number of other applications. And also trying to, on the flip side, trying to understand enough about adhesion that we can keep these things from sticking to stuff that we put in the marine environment that causes damage. And we call that biofouling.
So let's look at the two kind of range of applications, the positive uses of glues, as you said, potentially in surgery and elsewhere, and then how to deal with the problems of having a boat and it being fouled, as it were, with natural components of the ecosystems. How do we then use our knowledge about glues to deal with that? So what about the positive things?
So the positive things, I mean, we're getting, I think, far more aware of conserving our natural environment. This is something I haven't mentioned so far, but in order to develop marine protected areas and protect coral reefs and all the things that people are interested in doing, we need to understand something called larval supply. So there's no point protecting the adults that are already there. We need to understand how they get there in the first place. And about that, very little is known. It's incredibly complicated to research. There aren't many marine biologists working on that. So that's one of the positive reasons that I work on this. The other reason is, as I've already mentioned, trying to reduce the amount of nasty chemicals that go into synthetic glues. So if you look at things like plywood, plywood is about 60% glue, 40% wood. And until recently, those formulations have used highly toxic chemicals like phenol and urea and other kind of nasty chemicals. Increasingly, they're being moved to less toxic chemicals, which is great, but there are bio-inspired glues that are being used, and one of those is inspired by the adhesion system of mussels, a protein component called dopa, which is very sticky, which has been tested in plywood manufacturing and works and those types of things.
So you used the word inspired there. You're able to look at the mechanisms by which mussels and barnacles and sea anemones and such like attach, and then use our knowledge from that into a industrial or health or human application, as you were just describing.
Yeah, exactly. I mean, I'm a biologist, so I will always say we need to understand more about the biological system before we rush towards trying to translate what we know into a product. There's been an enormous amount of work in the last 10 or 20 years on trying to translate that knowledge, specifically about mussels, which is not my research, I should say, into products. But fundamentally, when you look at it, we know very little about that biological system, and I would say we need to understand more before we kind of invest large amounts of taxpayers' money in trying to take those principles that we do understand and find out that it's not the complete picture.
So track this biological knowledge into, as it were, in economic and industrial applications when we're thinking about anti-fouling and what boat owners and boat people need to do in order to create efficient craft, what's been the problem in the past and what could be revealed or inspired with this new understanding of adhesives.
So biofouling is probably the biggest economic and environmental challenge that no one's ever heard of. So, I mean, if you think about everything in this room, it's probably been on a container ship at some point. Containerized freight is the most efficient way of getting stuff around the world on boats. The problem is it's also incredibly polluting. So shipping produces about 3% of global greenhouse gas emissions, which is about the same as an economy the size of Germany. There's a study done by The Economist a few years ago that found that the 15 largest container ships in the world produce more nitric and sulfur oxides than all of the cars in the world combined. So it's an enormous issue. And this all comes down to drag. So if there was no hydrodynamic kind of drag of the water on the hull of the ship, you could give a ship a nudge in Tokyo and it'd drift across the Pacific and end up in San Francisco a few weeks later, assuming there was someone to steer it in the right direction. But that doesn't happen because of the frictional drag on the hull of the ship. And the more rough you make the hull of the ship, the more frictional drag there is, the more fuel the ship has to burn, and that produces greenhouse gas emissions and also costs an enormous amount of money. The fuel bill for a container ship can easily be $1 million a year. So when you think that a few barnacles here and there and some slime and a little bit of seaweed can increase the drag by about 80%, then these are staggeringly large numbers.
So we've got very large vessels which don't seem, from a distance, wouldn't seem to have a problem. But when you get close up to the dynamics of moving through the water, actually having algae and barnacles and other things on there is really important. So what has been the approach to that kind of anti-fouling in the past, and what does your research on adhesives and glues tell us now about what could be done?
Well, that's only one aspect of it, actually, because there are other challenges. One of the challenges is that by inadvertently moving species around the world on the hulls of ships, we introduce what we call invasive species sometimes, which can be environmentally damaging. And we still have areas of the planet, believe it or not, that are reasonably pristine, like Antarctica. And there's a lot of awareness now about, not moving things around. So you need effective, what we call anti-fouling coatings. And these have been used, since the time of the ancient Greeks and before in various ways, and they've not actually changed very much. We still, to this day, use a lot of heavy metals like copper and zinc, albeit formulated into expensive paints that we coat the hulls of ships with. And these kill things. That's how they work. They're biocides.
That's the idea.
Yeah. I mean, they work. They function by killing stuff. And apart from being kind of ethically challenging in this day and age, all of these materials, I mean, I often describe them as the only materials produced by humans that are designed to pollute the environment. I mean, they are designed to dissolve and release this into the marine environment, and they do so in enormous quantities. So we're trying to rein this in a little bit and think about how we can produce non-toxic coatings that interfere with the adhesion process of barnacles and mussels and all of these other problematic species. And for that, we need to understand how they stick in the first place.
Fascinating. Last year, you were one of 16 research groups from across the country who were selected by the Royal Society for their annual exhibition. Tell us a little bit about how that engagement with the public that came to the Royal Society to see what you were presenting them with, that interaction with the public and what you were telling them in that kind of exhibition that is about kind of public understanding of science, I think, isn't it?
Well, of course, we were one of 3 from the University of Essex, which was fantastic. It was fantastic from the point of view of raising the profile, but also from the point of view that the university was able to really get behind it. So we had an enormous amount of support, which turned it from something that could have been quite traumatizing, most probably, to something that was quite enjoyable.
Quite a lot of pressure. Yes.
So it was probably the most intense week in my professional career. Because actually, I'm not a natural kind of science communicator. As a lot of scientists are, my research is quite solitary. So getting out there and having those conversations with the public, it's fantastic, but it also takes quite a lot out of you if you're not a natural in those cases. And I suppose I'm not an evangelist about how my views of imposing my views on other people. I try to tell them about things that they didn't know already and use that perhaps as a nucleus for them to be curious about the natural world and look for things and think, if this is new to me, how many other things out there might we lose before we understand them? I mean, you don't meet, and with larval biology, it's very straightforward. I mean, you don't meet many members of the public who know that there are parasitic barnacles that castrate crabs and those types. Or if you do, you can have a great conversation about it and they don't think you're a weirdo.
Yeah. So the focus on that was a kind of alien environments and underwater, which we can't see, but all sorts of interesting things are happening. And it's fascinating how you can study specific organisms or parts of organisms' life cycles, and quite quickly that has applications that people can understand. You can talk about boats or wood or glues that may be able to help seal up bodies after surgery and so forth, and suddenly that brings it to life in a different kind of way.
Yeah, and the exhibit obviously wasn't only about my research. It was a multidisciplinary team from life sciences and also CFAS and other international collaborators as well. And it covered everything. It covered movement in the ocean in the broadest sense, so the movement of animals. And of course that spans from oceanic migrations of whales through the migrations of salmon for reproduction up and down rivers, and eels, of course, and all the way down to the micro scale where we have movement of single bacterial cells within biofilms. And all of these scales of movement are equally important. I would argue the small stuff underpins everything else, so arguably more important.
And so knowing about, as you were saying, that final thought about the need to protect marine environments more because of overfishing or other kinds of industrial damage suggests a need to know how environments are linked up to each other. So it's not going to be helpful just to protect one small patch. We've got to think about the whole system. And this research is telling us about larval movement and planning corridors of movement, or certainly connections, but thinking much more broadly than small locations.
Yeah, I mean, there's one ocean, effectively. It's all connected. And it's more connected now than it ever was before because of human activity. But also there are more barriers, physical and environmental.
So challenges are still there. Yeah. Good. Well, thanks very much indeed, Nick. Nick Aldred from the School of Life Sciences. You've been watching a Brighter Futures film. For more conversations on how ideas improve lives, pop along to our sister podcast, Louder Than Words.